It's my great pleasure to welcome Caroline Uhler from MIT to talk about mathematics of genome packing and regulation. Thank you for joining us today, Caroline.
Yeah, thank you all very much for inviting me. It's a pleasure to be here. So I thought I'll talk a little bit about geometric problems that have appeared when we have been studying genome packing and its effect on regulation. And so let me give you a little bit of an introduction of why we got interested in this problem. So as we all know, we have like all these very different cell types, and the expression in all these different cell types is actually very different to each other. But what is so interesting to me, at least, is, or what is such an interesting and intriguing question is, how is this possible since the information in every one of the cells is the same? So, you have the same DNA, which comes from mom and dad, that is packed into the cell nucleus and is packed into every one of our cells. So we have basically the same genetic information, this DNA, this 3-meter long strand, but it gives rise to all these very different cell types, muscle cells, neurons, etc. And they're all so different in their expression patterns and their function, etc. So how is this possible? And so it's becoming more and more clear that, the packing of the DNA really matters inside the cell nucleus, and that you want to bring genes together that are important for this particular cell type. So if you change the packing of the DNA, you can actually get very different expression patterns out, just because, you can bring different kinds of genes together that should be co-expressed in that particular cell type. And so we have been really excited about, studying the packing of the DNA, and this is, of course, a very geometric kind of question, for then understanding gene regulation down the street. And in terms of like data modalities, I think also what is really exciting in biology is that you can, study these single cells in many different modalities, in imaging, which is, a very nice modality actually to see how the DNA is packed. Also sequencing, you can actually also get to the packing of the DNA. We'll talk about that in high-C data sets. And then, of course, expression that you can measure in single-cell RNA-seq. And also something that I think is quite unique in biology is that you can intervene on the system in order to be able to understand gene regulation. So you can perturb the system in very easy ways. You can apply drugs to cells. You can use CRISPR to knock out particular genes and see how the expression changes, etc. So these are all kinds of handles that one has in order to actually get at gene regulation and connecting that to the spatial organization. So what I want to do is really try to study this interaction between, here on one hand you have the packing of the DNA, and on the other hand you have function. And as I said, there is really already quite a lot of evidence that the packing of the DNA really matters. Okay? So for in diseases, this packing can go off, and that can lead to different kinds of diseases. For the expression of particular cell types, you really see that particular genes have to come close in space. So what I want to do first is really concentrate on this packing aspect, and then I'll go the next step of like trying to connect the packing with gene expression. Show there how important, so the packing is of course already a geometric question. Then for connecting expression with the packing, we'll use a lot of autoencoders, and then want to talk in the second part on like the geometric questions that come up when we're actually using autoencoders. So these two geometry problems, so one packing of the DNA, and then the other one when we want to connect expression and packing through the use of autoencoders, and how important it is to actually understand the geometry there as well. Okay. So how do we go about actually understanding something about the packing of the DNA? So I'll be talking in this first part. There are different ways of studying. One can also study it using imaging data, as I said before. But first here, I'll be using high-C data, which is a really nice technique that has been developed in order to identify which regions of the DNA actually come close together. And so how this works is, we will be studying these kinds of matrices here. These are contact frequency matrices. And this is the technology to actually get this data, which is really quite amazing. But how you should read these kinds of matrices is, every entry here. So this is an average over many, many cells. Okay, so you take many cells and you just glue together everything that is close by through these, cross linkers here. Then you sequence them, and then you get to see, which region of the DNA was actually close to which other region of the DNA. And you just count how often would this particular region close to that one. And that's exactly what you get to see in these matrices. Okay, so this entry here would mean, in how many cells was this entry, this particular part of the DNA, found to be close together to this particular part of the DNA. So these are these high C matrices that we're going to be using. And from these high C matrices, we would like to reconstruct the whole packing of the DNA inside the cell. So this is the problem that I want to study at the beginning, and that has been studied a lot, and I already have here some references. So you have these contact frequencies of, this part of the DNA, how often and how many cells did it come close together to this part of the DNA. And from there, what you would like to do is actually be able to reconstruct the packing of the DNA inside the cell nucleus. Okay, so let me go with a particular approach that has been used a lot, and then look at, why is this maybe still not so easy as maybe people thought it would be. So a standard approach to do it is you take your contact frequencies, and we want to turn it into a Euclidean distance geometry problem. So we have our contact frequencies here. Frequencies have to be turned into distances, and people have looked at different kinds of ways of actually turning them into distances. One of them, and you can also do likelihood-based approaches where you actually try to figure out what this correct this correct function is, or the best function based on your data is, to actually transform these contact frequencies into distances. One of them is just to take this to the -1/2 here as a transformation, but you can also estimate this particular parameter up here. Okay, so let me just go with, you can either estimate or you can just assume some function that goes from contact frequencies to distances. And now once you have a distance matrix of every locus with every other locus, now you have a Euclidean distance problem, right? So I give you a noisy distance function of every point in the DNA with every other point, and I would like to find a 3D embedding that best captures these kinds of measured distances. And that has been applied, a lot here, and I will tell you what the problem is, but just, I'm sure probably everyone here is familiar with how this works, as how to solve a Euclidean distance problem. But just briefly, right, we know that if you have no noise in the data, and even if you have actually noise, there are also results on how many measurements you need in order to exactly be able to find the right embedding. But the standard thing is that you can solve this via a convex optimization problem. What you do is, here you have, so in our case, we want to embed our data into 3D. Say you have these n points, right? So this would be the, at what resolution you're going to look at your genome. So if we do it, say, a 250 KB resolution, we'll have about 10,000 of these little, nodes that we want, or points that we want to embed into 3D. So every one of these vectors would be a 3-dimensional vector. And instead of solving directly for the position of each one of the dots, what we'll do is we'll do this transformation into this gram matrix, right? Where, the entries of the gram matrix, so this II entry will just tell you how far away from the center is the ith dot. And then the Gij entries will tell you the angles between the two dots, right? So instead of having every one of our dots and its location, we'll just transform it into a problem of how far away you are from the center and the angles between any two dots. And that obviously also defines the whole conformation. And obviously you can only tell the conformation up to translation and rotation in space, but that's all we care about. And so if you do that kind of transformation, then you can write this out as a convex optimization problem. Here you're minimizing the trace of G of this matrix, of this gram matrix, because you want this in the end to be of rank 3, right? Because this is, if this is an embedding into R3, then this is a 3-dimensional vectors here. So this here is a rank 3 matrix. And so by minimizing the trace, you minimize the L1 norm of these eigenvalues. And so you want that to be sparse, right? You want many of them to be 0, and that's why, you can minimize it. Now, it has also been shown that actually maximizing the trace can also give you very good results, which is also quite interesting. But let's stick to minimizing the trace. Okay, so this is very standard, right? This has been studied to very, very, in very much detail, these Euclidean distance problems. Now, why is this maybe not yet solving the kind of problem that we actually need to solve in biology? Although this is the one that has been applied until now. But let me show you what is actually happening and why this is really not the solution that we're looking for. So as I said at the beginning, you get a copy of your DNA from mom and a copy of your DNA from dad. And the differences between the two copies are actually very small. So if you look at the DNA between any two people, most of it is actually the same. And there are just these SNPs, right, that differ between different kinds of people. And so if you don't have the exact SNPs on the whole genome, you'll not be able to tell the difference between a locus that comes from mom and a locus that comes from dad. Okay, and this is like kind of shown here in this little picture. So humans have 46 chromosomes, 23 come from mom and 23 come from dad. And so if you will tile this up into little regions, right, whatever your resolution is, let's say 250 kb resolution. And so now if you look at this first blue dot here, and this first one blue dot here, there's actually, there are now data sets where you can really tell them apart, but in general, when you take high-C data, there is no way for you to tell the difference between these. So xi and yi here, they are the same in this kind of data set, and for us, there is no way to tell the difference. So if you measure the distance between two nodes, it's really a sum of distances between 4 nodes. Okay, so if we take node I and node J, then say this is like one blue node and one red node here. In fact, what we're measuring is the contact frequency between xi and xj, between xi and yj, between xj and yi, and between yi and yj. Because we cannot determine, so they are indistinct, xi and yi are indistinguishable, and xj and yj are indistinguishable. Okay, so while we're getting and so this is what high-C measures, high-C would give you a n times n contact frequency matrix. But what you really want to reconstruct is 2 endpoints. Okay, so it's 2 endpoints sitting in R3 that you would like to reconstruct from an n times n distance matrix. So it's really a different kind of distance, a Euclidean distance problem that we're interested in. And so this has not been considered until now. So people would just reconstruct just half of the chromosomes. But, we know that if you do imaging studies, we know that it's not the case that you just have, that if you just look at half of the chromosomes and the other half, that the distances are just the same between them. There is actually an interesting packing that involves all 2n nodes, and not just n nodes and then a copy of these n nodes again. So we know that this is super important, and we know that we need to be able to get to these diploid packings and not just this haploid packing to really understand what is happening in the human genome. So this is the problem that we want to solve. So we're measuring this n times n distance matrix, or frequency matrix that will turn into a distance matrix. And then what we want to infer are these 2n points in R3. So it's 2n times 3 variables that we want to infer. And if we just do a little bit of a dimensionality count, right, it seems that there is at least hope to do it, because you have n choose 2 constraints and you have 2n times 3 variables. So, if n it doesn't even have to be that large, but then you'll have many more constraints than you have variables. Right? So there is at least hope that maybe we'll be able to do this. And the question is it identifiable? Like what we know for the standard Euclidean distance problem, we know that there is a unique solution. So is it identifiable here? And if not, what do we do about it? Okay, so that's the problem that I want to look at. Okay, so this is just putting it into mathematical terms, just so that these 2 points that are not identifiable between each other, one is called x and the other one is called y. Okay, so xi and yi, those are the ones that we can never identify. And then really the contact frequency between node I and node j is, as I said, it's a combination of 4 contact frequencies. And so we'll define these distances between nodes I and nodes j as these distances between 4 different nodes. Okay. So now the question is the 3D configuration identifiable? And if so, how can we compute it? Okay, so the first theorem that we have here is showing that it's actually not identifiable. Okay, so in 3D, if all you're measuring is these distances that are a sum of 4 distances, so we have this n times n contact frequency matrix that we are measuring, and every entry corresponds to these 4 distances added up. We would like to reconstruct this 3 times 2n variables. We can prove that this is not identifiable. In fact, there are infinitely many solutions, even though, right, just by a dimension and constraint argument, you would think that maybe it is identifiable. Well, these constraints are not independent of each other. And in fact, you can show that it is not identifiable. And there are infinitely many solutions. In 1D, it is identifiable. In 2D already, there are infinitely many solutions. 3D, it's not. It's, again, you have infinitely many solutions. And we also know up to what we can identify. And it's really quite interesting that what you can identify is you can, so for these 2 homologous, this is what they're called. These are these homologs, right? The one that comes from mom and the one that comes from dad. So this xi and yi, these are homolog pairs. The xj and yj, those are homolog pairs. What is actually quite interesting is that you can identify the distance between them. So this thing that you cannot measure is the distance between them, but this is identifiable. What that non-identifiability comes from is that these 2 pairs, they can be anywhere on a sphere. Okay, so they're anywhere across from each other on the sphere. So that's the non-identifiability that you have. So there are infinitely many solutions, and of course also in 2D there are, because then they're all on a circle. Okay? So you have identifiability, you have unidentifiability, and the unidentifiability is for any homolog pair, it's on a sphere, or in 2D on a circle. So that's kind of disappointing, right? Because people have been measuring these high C data sets and thinking that one can actually identify the packing of the DNA. What we're showing here is that you can actually not do that. Even if you don't have any noise whatsoever, you cannot do that. Okay, so then, we don't want to stop there, right? We do prefer positive results than negative results. So let's think about what other data could maybe be used in order to actually be able to identify the packing of the DNA. And so if you have any questions, just let me know. Okay, so otherwise I'll continue. So there are other kinds of constraints that you can use. And in particular, right, if we look at the DNA, well, it's not just points, right? So we know that things on a chromosome, we know that this point comes after this point on the chromosome. They are one after the other. So we should be able to tell the distances between them in 1D. And so we could add in distances between these neighboring loci, as you see here. So if you have here a dot and then here a dot, depending on the resolution, you'll be able to tell how much this distance should be in average, right? So you actually have quite good measurements of this. If your loci are not too huge, right, then whether it's curved or not curved, this actually doesn't matter as much. And so you could add in these constraints between neighboring loci. And in fact, we can show that that gives you identifiability in 2D. It still doesn't give you identifiability in 3D. So what can you do in 3D? And so here is something that I'm very excited about, these new data modalities that are coming out and these technologies that are being developed. So since, all these examples that I also already showed at the beginning, it is clear that what matters is really spatial organization of the DNA. And it's not just pairs of genes that come close in space that are functional, but even actually multiple genes that are coming close in space and are functional. And so there are these technologies that are measuring these higher order contact frequencies, where they measure how often do these 3 points on the DNA actually come close together, and not just pairs of things like how we saw in high C. So the output of, for example, Sprite and other technologies is how often do these different kinds of points come close in space. And that's what we're going to use, and actually show that with that kind of data, you do get identifiability. So now we first need to set up the problem, and I think here there is some choices to be done. But let me tell you the kinds of choices we made, of course, because we want to, in the end, be able to turn it into a convex optimization problem as well. Okay, so we need to somehow turn higher dimensional contact frequencies into distances. And for sure, here there are other choices to be made. And again, as I said, this is one choice that we'll be making. Under this choice, we can show that we get identifiability. It is an interesting question to, think through what could other choices be, and would this also lead to identifiability, etc. But let me explain what we did. Okay, so we need to go from contact frequencies to distances. So how would we do this? Well, so contact frequency, so let's first look at the haploid setting when we don't have 2 copies of every chromosome. So in the haploid setting, how would we do this? Well, so 3 points have to come close together. That's what we're measuring in the contact frequency land. Well, how would we turn this into distances? What is an intuitive way of turning it into distances is, since these 3 things have to come together, well, what matters is the distance. So how often the 3 things will come together depends on the distance of all these things to the centroid of, these 3 points. And so how we'll turn it into distances is by just, the sum of the distances of every one of these dots to the centroid. So this is the function that we're going to be using of going, and then to the -1/2 or whatever the parameter is that you want to be using. Okay. So this is the function that we'll be using in order to go from contact frequencies to distances. That's, kind of arbitrary. I think it is intuitive that what you want is the center, is maybe the centroid of the 3, because you really care about all 3 of these points coming together. But again, you could use something like a likelihood-based approach where you actually try to estimate what this function is. But this is what we used in order to then try to analyze whether now we actually get identifiability or not. Now we still have to do this for homolog chromosomes, right? So again, we have 2 copies of every one of our nodes. So how would we do this? Well, so before, right, when we were looking at the distance between 2 nodes, that gave us the sum of 4 things to look at. So now here, if you look at 3 nodes, so that now suddenly gives you 8 different kinds of configurations to look at. So for every one of a choice of these 3 loci, we'll get 1 centroid. And now we need to turn frequencies into distances, so it cannot be the sum of them, right? Because then, so what you're measuring is really the sum of all these frequencies of these 3 things. So you can certainly not take the sum of the distances, because then bigger frequencies will correspond to bigger distances, which is certainly not the case. So what we did, and again, this is like somewhat arbitrary, but I think it biologically is an assumption that is, if you look at imaging data, it might be a reasonable assumption, is that we said that, well, among all these 8 possibilities of things coming close together, there will be one that is dominant. And so we'll just take the minimum over all these. So we have 8 distances that are defined here, and we'll just use one of them, namely just a minimum distance. That is going to be the triple that will give rise to, the biggest part of all of these frequencies. And so that's the one that we'll be using. And so that's what you see here. Okay, so this minimum here. So you have, 8 possible, if you're looking at triples of things coming together. Of course, if you're looking at 4 things coming together, you'll even have more possibilities. So here, if we're looking at triples of loci coming together, when you have, the diploid setting, you have 8 possible triples coming together. And we'll just say that, one of them is usually closer than all the others and will be the one that gives rise to the biggest amount of the frequency. Everything else will just will be negligible, will not take it into account. And so we'll just take into account one of these 8 pairs. All the others have such large distances that they will only contribute very little to the frequency, and hence we won't take it into it. That's also for computational reasons, and in particular, we'll not just look at triples, but we'll also look at 4 things coming together, 5 things coming together, etc. All of that is in the data set. So this is the kind of assumption or simplifying assumption that we made. We just wanted to understand whether these kinds of assumptions or this kind of data could then actually lead to identifiability. Okay, so after you make an assumption of how you actually want to transform frequencies to distances, then now you can ask, do you actually now get identifiability? So as I already described before, so just adding these kinds of neighboring distance constraints already gives you identifiability in 2D. So we have a proof for that. So now the question is in 3D, do we actually get identifiability when we add these higher order distance constraints into the problem? And what is really quite nice is that we do. So what is the theorem saying is that, so we have these high C constraints, so these pairwise distance constraints that we had at the beginning, where we know that we can identify up to a sphere of any of the homolog pairs. We add the distances between neighboring genomic regions or loci. We know that that gives you identifiability in 2D, but not in 3D. And we add, and in fact, here we're just in the theorem, all you need is the third order tensor constraint. So just the distances between 3 pairs. And in fact, you can even say how many of them you need. And you need that at least the first and the last entry of each one of the chromosomes is included in some of these 3-way constraints. Then that's enough to anchor the full packing of the DNA inside the cell nucleus. So under these, when you have these 3 measurements, then in fact you do get unique identifiability of the DNA packing inside the cell nucleus. So it's interesting that these higher order constraints actually really help you in this. Okay, so let's look at how, and then of course, okay, so all these constraints are nice, now all you have to do is take this and do whatever we did in the standard, or whatever has been done, it's not our word, and has been used a lot for the Euclidean distance problem, right, of going from this, using the variables xi, which are the coordinates of these points, turning that into a gram matrix description, and just writing all of these distance matrix constraints in terms of the gram matrix. And it's really nice that you can actually do that and write it out again as an SDP. That's exactly what we did using these now additional constraints, and then just solve it. And so here I'm showing you some simulation results. So, if you have no noise, then since our theorems say that we should be able to identify it uniquely, we should be able to identify it uniquely. And that's exactly what you see here. So I think, I don't remember which one was which, but I think the dotted line is the reconstruction, or is the real one, and the straight line is the reconstructed or the real one. And you see that you have different models for generating data. This is just randomly generated data in 3D, and no matter what you do, right, you will be able to reconstruct this. So that's nice. So at least the theorems actually hold true. And so what about when we're now trying to analyze real data? So there is the Sprite data set. So this is the one that we used on the same cell type. So you use a Sprite data set, which has 2-way constraints, and then also 3-way and higher order constraints. We only use the 3-way constraints here. The other ones were really quite sparse, in fact, in terms of how often they're measuring higher order constraints, but you could add them in. It's just computationally a problem. So here you see the whole packing of the DNA. Inside the cell nucleus, so with all of the chromosomes, and always 2 copies of each chromosome, so how they actually happen in the human. And then here is just a little bit of an analysis that we're actually getting something reasonable out. So here are some trends that are well known. So in particular, what is very well known is, and so now hopefully I'm reading this correctly. So what we know, and then we should plot it back to the picture, is that the small chromosomes, they tend to be in the center, whereas the bigger chromosomes tend to be outside. And so that's exactly what you see here. So the small chromosomes, mean chromosome size, they tend to be more in the center, and the bigger chromosomes, they tend to be more outside. And you also see something in terms of activity. So this is usually measured by these A compartments. You see that the very active loci, they tend to be more in the center, whereas the less active loci tend to be outside. And that's also something that is actually recapitulated in this kind of packing. Although, of course, we never, add in any information about how much locus is expressed or not. This is in fact just coming out from this high C, from the Sprite and from the Sprite data that we get in terms of the packing that we actually identify. Okay, so I think there are quite a lot of really nice geometric questions that appear when you're trying to do, trying to infer the packing of the DNA from different kinds of data sets. We did it here for this average data set. I think there is a really interesting kind of geometric question also in single cells. So there is now single cell high C data where you get to see every locus. And again, you have the same problem, right? That every contact. So now it's just 0 or 1 or, sometimes 2, well, but usually it's just 0 or 1. It's a binary matrix where you get to see whether 2 loci actually come in contact or not in the single cell, so not averaged or not added up over many cells. And so then the question is, right, if you have the 0, 1 matrix where a 1 could correspond to any of these 4 partners coming close together, well, how many of these contexts would you need in order to identify really this packing within this single cell? So that's a rigidity kind of question, and would be an interesting question to look at as a next step, which we have not looked at all. Okay, so at the single cell level, it leads to very different kinds of questions. It's like a graph rigidity kind of question that you want to analyze there. Okay, so those are the kinds of geometry problems that I think are really exciting when you're looking at the packing of the DNA. So that was all still on this side of the spectrum. Now what we're really interested in our lab is to connect the packing of the DNA to actually gene expression and gene methylation. And so for this, I'll be introducing these autoencoders because we've done a lot of work using autoencoders to do this, to really combine like two very different spaces with each other. And then I'll get into instead of like a geometry question that comes directly in the biology, I'll get to a geometry question that we need to understand in order to then, about autoencoders in order to then say something about the biology. Okay, so this is kind of shown here. So we care about the packing, but we're going to use these autoencoders, which are these neural networks, and we want to understand something geometric about them in order to then be able to say something interesting about biologically. Okay, so let me explain what I mean by this, and let me just show the full slide and introduce these autoencoders. So if I go back again, so why am I looking at generative modeling, and why am I looking at autoencoders at all? So what I would like to do is, you give me something like the packing of the DNA, and I'm the single cell level, and say we will be measuring this now with imaging. So we don't know where each gene is, but we'll just get to see the whole. So we'll just fluorescently label the whole DNA, and so you'll get to see where is the DNA densely packed and where is it not so densely packed. And then from this image at the single cell level, we would like to be able to predict the whole gene expression pattern at that single cell level. So that tells you that we need something like generative modeling because we will want to generate from an image, we want to generate the expression pattern of that cell. So we need, generative modeling for doing this. And let me just introduce this because probably most people are more familiar with discriminative modeling and neural network use. So, discriminative modeling is you have training data that is labeled like cats and dogs, right? And often you just train a neural network to figure out a good embedding where you can actually distinguish very well between cats and dogs. Now, that's not what we have. We don't have any labeled data, etc. And we want to generate like other kinds of data sets. So what we're looking at is this other area in machine learning, which is generative modeling, where, you just have examples, not necessarily labeled in general. And you want to find a good embedding of these examples so that you can generate new ones. And so what would it mean to kind of be a good embedding? Well, it could be a good embedding because if you cluster the data and the embedding, you actually find out that, here are the cats and here are the dogs and here are the rabbits. And a good embedding is also one where you can then generate new ones, as I just said. In particular, for example, here, if you would walk from rabbit land into cat land, and you go here and you generate the corresponding image corresponding to it, maybe you get out something like this, like a cabbit, a mix of a rabbit and a cat. Okay, so that would be a very informative kind of embedding where you can actually generate stuff that actually is meaningful. And so how do people do this? How do people get these informative embeddings that are, you could also classify in this embedding, but it's not just optimized just to classify. It will be informative for also getting clusters out or generating new data, as I just. So how do people do this? And this is what we're going to be analyzing, are these autoencoders. So what you do, again, you don't have any labeled data, so you just have a mix of, say, images or sequencing data, or whatever it is. And that's your training data. And so now, so this is a generalization of PCA, and it consists of two neural networks. So one neural network that goes from, and let me just do the example with imaging data. It goes from your imaging data to some latent space, which is, the space where we're then going to do all the clustering, etc. To this latent space. Then you have another neural network, which is known the decoder, which goes back to your original space. So if it's images you put in, images will come out of the same dimension. Okay, and so now you have your training data, and how do you train all of these parameters in this neural network? Well, you'll just train, so you want to find a representation here that is as informative as possible. So how you train it is just so that, whatever you put in should also come out. Okay, so you'll train it to minimize reconstruction error so that, the cat that you're putting in, also a very similar cat will come out on the other side. And that should give you a representation in the latent space that contains as much information as possible, right? Because it has to contain so much information that you're actually able to reconstruct whatever you have put in. So that's what an autoencoder does. So it's a nonlinear version of, say, something like PCA, right, where you also find an embedding that is as informative as possible, but using something linear. And here you can use something very non. Okay, so these autoencoders have been used a lot, and I'll show you on the next slide how we're using autoencoders to actually solve this problem of going between different data modalities of the images in, the sequences out. And what I also want to say is like how these autoencoders can be used, and this will be also important for actually understanding or to lead to these geometric kinds of questions, are these questions of style transfer. And again, I'll show you how we're using it in biology and why, this is like, so these autoencoders have played such an important role and have been really, really important for our lab's work in biology. So here is a question that, of course, has been studied a lot in vision. And I'm sure you've seen this, like these pictures of Yosemite in the summer and then the same picture in the winter, where you do the style transfer between summer and winter. Or you can do this kind of style transfer that is shown here. So say you have, many pictures of people. And some people you have in this data set that are in a neutral face, and sometimes they're in a smiley face. And so now you have here a new person, and this person, you only have this person in a neutral face, and you would like to generate a picture that corresponds to this person smiling. So what you do is, again, you train, given all of your data that you have. You don't have this person smiling, but with all of the other data, you just train your autoencoder, and you'll get this latent space representation that we have also on this side here. And now what actually works quite well, and it's quite surprising, and it's a really interesting geometric question, is, take a person that where you have it in neutral face and in smiley face, take this vector here. Okay, this corresponds to adding a smile, because this point here is a person in neutral face. This point here is a person with a smiley face. Take this vector and just move it over to where the person is who is not smiling yet. You add this vector here. You move this point back to the image land, right? You just decode it back to images. Like here, I can move around, and then this point here, I can actually map it back to images. Out comes an image, and out comes an image of this person actually smiling. Now, this only works if, your latent space has very nice geometric properties, right? That adding a smile is actually something that you can just move around, and no matter where you're going to add it, it will be adding smiles to people. And so the question is, of course, why do these latent spaces become so well aligned? And so let me show you how this can be used in biology, and that they actually do become nicely aligned for at least certain applications and for certain kinds of autoencoders. But first, let me show you the example of how we use this, how we use autoencoders in order to go between very different data modalities. So I told you the thing that what we're going to measure are images. So these are the real images of the packing of the DNA. So just fluorescently labeled DNA. And what we would like to generate is actually the expression. So you give me a particular cell, and I tell you what its expression profile is. Difficulty is that you're never able to measure both at the same time, because actually getting an image means destroying a cell. Getting an expression pattern means destroying a cell. So you're never able to measure the same cell, so you don't have paired data, and still you want to be able to somehow pair these data so that I can actually generate the other data modality that I haven't seen. So the question is, how do you do this? Now, let me go through this the little example. So how we're going to do it is you take a full population of cells, say these are T cells, you take out some for imaging, you take out some for sequencing. So it should be the same population of cells. So in some sense, in the intrinsic state of cells, they should have the same distribution, because I just took out some at random for sequencing and some at random for imaging. So there should be a latent space where the two distributions are actually the same, because intrinsically it is the same population of cells. And so that's exactly what we're going to try to do. So you have here, and you can have two modalities, you can have many more modalities. You have one autoencoder that goes from images to latent space back to images, like what we had here. Images to latent space to images. You can also have autoencoders that go from sequences to latent space to sequences. So we'll have other autoencoders that go from sequences to the same latent space to sequences. And now what we need to make sure in the latent space is that the two distributions are aligned. And how you do that, and this is something that is quite nice in machine learning, is you just try to make another neural network that can discriminate. So if I'm in the latent space and I can tell whether a new point that comes in comes from imaging land or whether it comes from sequencing land, then I'm going to be punished, because then my embedding is not good. right? Because I at random took some cells for sequencing and some cells for imaging. So in the latent space, there should be no difference between the distributions. I should not be able to tell whether a sample comes from imaging land or from sequencing land. And so that's what you can just enforce in the latent space, that you can actually not tell the difference between these two distributions apart, which will make the two distributions. And so that's exactly what we did, and also validated that this works. But just so that you have a little bit of intuition, how this actually looks like. So your data modalities can also be different imaging data sets. So here, you can take this celebrity data set and so you can make, like, say, one data modality can be black-haired females. Then the other one can be blonde-haired females. The other one can be black-haired males. The other one can be brown-haired males. This now could be all my data modalities. And so if I train this autoencoder here, then what you can do with it is I can take a black-haired woman and move her to the latent space and generate what she would have looked like were she blonde. And so that's exactly what we're doing over here. Okay, so just to make it clear, so all of these here are generated images. They're not real images. All you're putting in all of these here are the real images. And all of these here are generated how they would have looked like were they a blonde male or a black-haired male, etc. Okay, that's just to show you how this works, because it's more intuitive on imaging land than if we do it on sequencing. So here we're taking in a real image and we're actually generating how it would have looked like were I also able to get its expression pattern. And the other way around here as well. And then we validated this. I said you can never measure everything paired, but you can in an image also measure, particular proteins. And so we're just looking at particular genes or particular expression profiles that can distinguish between two different states, also color them inside the same image, and be able to show that we can actually generate this correctly. So the amount of these proteins as well. So that's what I mean by validation. Okay, so you see how important it has been for us to actually, use autoencoders, and that's why it's, I think, really important to actually study these autoencoders. And here is also the second application that I mentioned about this alignment. Again, just showing you that these autoencoders are super important. What we wanted to do here, and we applied this for SARS-CoV-2 drug repurposing, is, you have all these data sets, and data sets like the CMAP, where you have many, many drugs or other kinds of perturbations applied to, cancer cell lines. And now, of course, this is a huge effort to do something like this. But now, say you want to do something like figure out what these drugs would do on for a different disease and a different cell type. Now, drugs can have very different effects, whether they're applied to a skin cell or a muscle cell or a neuron. And so now if you care about SARS-CoV-2 infection, well, then you care about lung epithelial cells that are infected by SARS-CoV-2. And so now you would want to be able to predict what will happen when you apply these drugs to this particular cell type that I have not measured in this data set, right? All that has been measured are all these different cancer cell lines. So how can you do this? Well, you could consider it as a style. So you have here your different cell types. So for this cancer cell type, I do know what the effect is of drug A. I have here my SARS-CoV-2 infected lung epithelial cell. I would like to predict what the effect is of all these drugs, and then I'll just, rank the drugs based on how much they're going to push me back to the normal state. Okay, so you want to rank these drugs based on how much they're going to push you back to the normal state. So I want to really be able to predict this. And what we showed in this paper, and so of course you can validate it using the data that you have here, right? You can take like two different cell types, and you can look at whether the effect of the drug is actually aligned in the latent space, because only if it's aligned, as I said before, can you do this. Can you actually, take the effect of a drug and move it over and then just be able to predict what this drug will do. And what we showed, and this is where, then the mathematical questions and the geometry comes in, is the following. So, and this is maybe unintuitive at the beginning, but I'll at least give you a little bit of intuition in the, say, last 5 minutes, and this will also lead to all kinds of open problems. Is the following. So autoencoders usually are used in this way, where you go from some high, like PCA, you want to find a lower dimensional embedding that is informative. So you go from your high dimensional space, the latent space is something lower dimensional, and you go back to your higher dimensional space. And you want to find this low dimensional embedding that is as informative as possible. Now, and I'll show you the theory on the next slide. So we'll do something crazy, and there will be theory for backing this up, and this is really also where the interesting geometry questions come in. So instead of going to something lower dimensional, we're going to propose to go something higher dimensional. Okay, so I have my original data, In this case, all these data sets, they live in 1000 dimensional space. It's known as L1000 data. So they all live in this 911 dimensional space, so around 1000 dimensional space. Instead of going to something lower dimensional, I'll go to something higher dimensional. So I'll embed them in a higher dimensional space. And then, move it back to the lower dimensional space. Now you can tell me this is completely crazy because this is, over-parameterized. So in general, I could just learn the identity map, right? I could just have the identity map go through here. And, that would give me 0 training error. And, but it certainly wouldn't give me anything interesting because then I could just have done it in the original space instead of this higher dimensional space. Now I'll show you on the next slides that, this is in fact not learning the identity map, and something very interesting is learned by this autoencoder. But what we showed in this paper, and this was, a very, this was a SARS-CoV-2 paper, but in there is the motivation for really that this is the right thing, is that if you look at how aligned these effects of the drugs are, and in fact, I know that pharma industry has done this and have shown that this doesn't work. If you just do that in the original space or using your standard autoencoder that goes to a lower dimensional space, these vectors of the drugs are not aligned. Okay, they're in general just not aligned. I mean, it's not the case that drug A, you can just move it over to a new cell type and then in fact predict what will happen to that cell type. When you use these over-parameterized autoencoders, they are in fact aligned. Okay, and by that I mean much better than what they were in the original space, or even definitely much, much better than what they are in this autoencoder space. So if you're doing this embedding into a larger dimensional space, it will make your effects of drugs be more aligned. And so the question is, of course, why is this happening? And we don't have the answer for exactly this, but I'll at least want to, in these last, say, 5 minutes before, I guess I don't know when I should stop for questions, but let's say in 5 minutes, when, what is the intuition for actually even using these over-parameterized autoencoders? And then we really need to understand why this is happening. At least we have some intuition for it, but we certainly don't have a proof. But I'll tell you at least that they're not learning the identity map and go through a little bit of math of why they're not learning the identity map and saying that they're actually really learning something very interesting. And we definitely have to understand it because they're just so powerful. They're so powerful for predicting the effect of drugs. They're so powerful for moving between very different kinds of data modalities, etc., that we really, really need to understand these autoencoders, in particular in the over-parameterized. Okay, so let's go through a little bit of math. Okay, so what I said, what these autoencoders do, how you train them, is you just try to minimize reconstruction error, right? So of what you put in, same thing should come out. So let's do the over-parameterized setting where, say you have less samples even than the number of variables, right? And so here is what I said, like this psi here is just the autoencoder map. Okay, so whatever you put in is xi. You have n training examples, outcomes psi of xi. What you're trying to minimize is this reconstruction error. So let's just, to get some intuition, let's just do the linear setting. And let's just look at one training example. Okay? So let's do one training example. So then what you're trying to minimize is, a times x minus x into norm, right? Over all a's. Over all a's and say RD. Okay. So now we all know, right, this has a lot of solutions. In particular, A could be the identity map. That would give me 0 training error. But A could also be a rank 1 projection onto my training example. It could be rank 2, it could be rank 3, etc. It could be full rank, which would be the identity map. So there are many, many solutions to actually trying to solve this optimization problem. And so the question is, what does an autoencoder learn, right? Which one of these solutions is the autoencoder actually learning? There are many, many solutions, but which one is the one that is being learned? And so we know this from regression. I mean, this is actually quite easy to prove, that if you just do gradient descent, and neural networks usually you start, so this depends on initialization of how you're initializing A. These are the parameters in this neural network. And usually you initialize them as close as possible to 0. If you initialize at 0, you cannot move. So you initialize them as close as possible to 0. In your proof, you just say, okay, I initialize at 0. And then you can easily prove what will happen. And in fact, the solution that you're going to get at is the rank 1 solution. Okay, so now that's quite interesting. So let's look what the neural network. So in the linear setting, with one training example, we know that we should get the rank 1 solution. So let's just actually run, these standard autoencoders, like these units with a lot of non-linearities and very deep ones, etc. Let's run them on one training example and see what happens. And you see that what is really quite interesting, what happens is that you train on one training examples, these rabbits, no matter what you put in, the rabbit comes out. So it didn't just learn the rank 1 solution, it actually learned the point map that, whatever you put in, the same example comes out. Okay, so that's a special to nonlinearity, because if you had just a linear network, in fact, you could also get out the negative of the training example, right? It would be a linear combination of the training example and 0, so it could also give you the negative of this training example. So, but if with an autoencoder, what you get out is in fact the point map, which is really quite interesting. And again, I should say, you should be surprised by this because there are infinitely many solutions, and the neural network, this is the one that it is learning. And in fact, as I said, if you're in the linear setting, it will learn a linear combination of your training examples. It will not learn the exact training example. And also, if you're not very deep, it will in fact do what people have known that autoencoders do. It will usually give out something. So even if you train on one training example, it will give out something that is similar to whatever you put in. Okay, so here you put in this example, out comes something that looks very similar to it here as well. So it's not the case that just a rabbit comes in. So it depends on the nonlinearity, and it does depend on the depth. And so the question is, even if I look at this, and there have been papers that say like autoencoders learn the identity map because they've seen these kinds of behaviors here. But in fact, we can prove they don't learn the identity map. And let me, and this should give you the intuition for these over-parameterized settings. They don't learn the identity map, and how do we prove that they don't learn the identity map? And where the geometry comes in is I can just iterate the map, right? Because an autoencoder maps you from RD to RD. So if you put in an image, another image comes out. So let me just put this image back in and see what happens if I just iterate the map. Well, so this will show you something quite interesting. So if you iterate the map, so here we trained our autoencoder, we put in images that have, we just added in a lot of noise on top of it. We just iterated the map. So after one iteration, this is already after many iterations, after one iteration, something comes out that looks, very similar to it. You just keep iterating. And then what you see is that you're actually going to converge to one of your training examples. So these neural networks, so it looks like the identity map here, looks like the identity map if you're not deep. If you're very deep, then this happens, like what we showed here, that, immediately a training example comes out. But if you're not so deep, in fact, if you're very, very shallow, well, it looks kind of like the identity map, but you just keep iterating and you can analyze what the map is. And you'll see that the map is super attractive at the training examples. In fact, we have not found any other attractors than these training examples. We cannot prove that, but if you visualize it, also this is quite interesting. I think this is where the important geometry will come in. We really need to understand these basins of attraction. So what are these basins of attraction that are learned, right? Which images are being made more similar? For our drugs, we need to know why are these over-parameterized settings making drugs that we have already measured more similar to each other? That's what is happening. It's like the alignment, right? It's aligning drugs that are more similar to each other to make them more similar to each other. It's like here what is happening is if you have an image that looks similar to this image, well, this autoencoder is going to map it to something that is even more similar to it. And if you iterate it many times, well, it will actually map it to one of these training images. That's at least where our intuition came from, to actually look at these over-parameterized settings. But we have no theory yet. So, okay, I should show you the theorem. So we do have a theorem that says that this attractiveness actually happens if you have one training example. So if you have one training example, no matter what your nonlinearity is and your initialization, I can find you the depth and the width so that you will be actually, you will be attractive at this training example. So that's the proof that we have. We don't have it for multiple training examples. We only have, all of these kinds of simulations that we've done with different kinds of nonlinearities, etc., different kinds of optimizers. All of them have exactly the same phenomenon of always converging to your training example. We have large networks where we don't find any other attractors other than our training examples. So no matter what you do, you're always converging to one of your training examples. What we don't understand, and I think this is the really important geometric question, is what are these basins of attraction? So where are the boundaries, right? How is the space broken up? How does this autoencoder decide who is more similar to me and who will be mapped to me in these tractor landscapes that are actually being learned. That will be really important for the different kinds of applications, because we're relying on this when we're doing, when we see that our autoencoders work well, and we need to know that, different nonlinearities will actually give you different kinds of basins of attraction. So depending on how you choose your nonlinearity in the autoencoder, you'll get different kinds of these. And so there should be some better ones for different kinds of applications and worse ones for other kinds of applications. So that's the kind of open question that I want to leave you with. And maybe I'll actually go, so that I have at least some time, I'll go to the conclusions and maybe just point out open questions. So I did talk about, this chromosome packing question at the beginning, then talked about autoencoders, showed you how super useful they are for all kinds of different biological applications, how important over-parameterization is to actually get things to be aligned with each other. And then these questions of that we really need to understand kind of these autoencoder basins of attraction that are happening. And what I said is, if you use different kinds of nonlinearities, you can actually get very different kinds of basins of attraction. We can visualize them in 2D. We cannot, of course, visualize them in very high dimensional spaces. But you see that you get very different basins of attraction, and it will be super important to understand them so that we can choose nonlinearities that are actually aligned with the kind of application that you're working on. And this will be super important if we want to get even better kind of alignment of different kinds of drugs in this latent space. Okay, so with that, let me end and really thank, amazing students. It wouldn't have been possible without them being just really, really forward-looking and doing all of this really amazing work. Then my collaborators on the biological side, everything I've done is always in collaborator with G.V. Shivashankar's lab at ETH Zurich, and then the over-parameterized paper on the theory side of, that these autoencoders learn they learn maps that are attractive at the training examples was in collaboration with Misha Belkin at UCSD. And then, of course, I want to thank all my funding sources, and end with an advertisement that we have a new center at the Broad Institute, and are always looking for really very talented computational people and people at the interface between any kinds of areas in mathematics and biology. Thank you.
Thank you very much for a fascinating talk. Really, really interesting. Biology, machine learning. Do we have any questions from the audience? Please speak up if you have some questions for Caroline.
Yes, Caroline, it's DeWitt Sumners here. I had a question about when you go up in dimension, how far up do you go?
Oh, this is great. Yeah. So actually, even if we're just a little bit over parameter, so it depends on if you're asking me about the theorem of, how far up do we have to go so that we get this attractor landscape. And there, per nonlinearity and initialization, we do have a theorem which tells you exactly how far up you have to go in order to make your training examples fixed points, attractive fixed points. If you're talking about the applications, actually just a little bit of over-parameterization is enough. And the interesting thing is, kind of how you should see, what your over-parameterization means is, if we're super, super over-parameterized, then kind of just once applying your map will already give you, you'll just jump direct. So if we're looking at this kinds of pictures, in one step from whatever you put in, you'll jump to your next training example. If you're not super over-parameterized, then you need many steps until you're actually going to jump to your training example. And so for applications, we want to be very little over-parameterized, right? Still over-parameterized so that at least it's attractive at the training examples. Not super, because you don't want to just get out the training example. You still want to be learning something interesting in terms of these maps.
Thank you.
Yep. Okay. Any other questions? While we wait for other people to come up with questions, I have one, and the slide that you're showing is really perfect. Do you know if anybody has looked into really studying the shapes of these borders between the various zones? No, because this is a paper that came out at the end of last year, where we first showed that there are these autoencoder basins of attraction. So no, and I think it's a really important open problem. Yeah, because I know that people have started applying methods of TDA to studying decision boundaries for machine learning in general. So it could be really interesting to see whether some of those methods could be useful in this context, I think.
Yep, and I would love, that's why I'm presenting it here. I would love for people to actually study these and figure out how they change with different kinds of nonlinearities. Yep.
Great.
Yeah, so here we're just showing that this is happening and that someone should look at what these are.
Pointing out, hey, this looks interesting. So, yes. Anybody else have a question?
I have a question for Caroline.
Go
ahead, Christian. Go ahead.
My name is Christian Micheletti. Pleased to meet you, Caroline. It was an excellent talk. I wish to ask you about the first part of the talk regarding the 3D reconstruction, let's say, the spatial reconstruction of chromosomes. So aside from the problem posed by the fact that you have double copies of chromosomes in eukaryotes, in humans, for example, there is somehow a more, maybe even more pervasive problem of heterogeneity that is really intrinsic to bulk high-C data, and that is the fact that the measurements, as you know, are collected over ensembles of cells. So there could be a different cellular stages, and so essentially what you measure as an intensity in the high-C map is actually the results of data from proximal loci collected over a huge ensemble. And so that is even more pervasive, as a problem. So what the intensity is not much as a distance, but an average ensemble of cases where you are in close proximity, where you are far away, you are never close, you are always far away, whatever. And so the way that polymerists have dealt with this problem is by taking into account the chain connectivity as a constraint in models. So you start with filaments that are somehow packed in some reasonable starting way, and then impose as distance restraints the proximity of pairs that are significantly above the background in terms of noise, which is very different from searching from a one structure, one set of distances. So can you comment on this inherent heterogeneity?
Yeah, very happy to. I mean, that's why I also asked the question in the end. Like you can ask the same question in single cell. There is single cell high C, right? And then you only have one cell and you're not actually summing up over many things. But in some sense, this already shows you that even if we have, and this is, I think, is the same problem for polymer physics models, and they will try to output many different configurations. But what we're showing here is that if you just have these measurements, and even if you're able to get these measurements, you have an infinite number of configurations that satisfy them. So that to me means that, I also don't trust the polymer physics kinds of models, because, even if I have these perfect measurements and yes, I know there's even more heterogeneity than what I'm doing in here, right? I'm just assuming that I have everything perfectly measured, and even then I get an infinite number of configurations out, and I cannot identify it up to a rotation on a sphere. So that just says like how under constraint this problem is. And that one needs to use other kinds of data modalities, whether it's in a physics model or whether it is trying to just reconstruct one of these. But I think, as you're saying, it might be really good to just start with single cell high C data and have a theorem which says like, now I get a binary matrix. I know these two regions are together. I know these two regions are together. These two regions are together. Can you now prove how many of these do we need and how many constraints do I need? And is it actually possible to say like, yes, this will define the full 3D packing of the DNA? But yeah, I agree with you. I think this just shows that, it's super under constraint.
Thank you.
Yep.
Other questions?
Well, Renzo Rica here, Caroline. I have a very superficial question, and this at the beginning of your talk, you showed the interesting search for a relation between packing and function. So the question for me is, do you have any information from what you've done so far, what has been done so far, about the density, the packing density, and some fundamental functions? And in particular, you show the diagram as an example where you showed the what you call the A compartments in relation to radius, to distance from the center. And the second question is that some kind of generic feature that you have a kind of a spherical symmetry where you can identify region like a shell model for your for your system?
Yes, so these are all great questions and questions of great interest in biology. And so, yes, so it has been shown that, if you want to shut down DNA and have these genes not be expressed, what you do is you just pack them very densely. So no transcription factor can go in. You don't care about these regions. So these are these repressed regions, and you can measure them with other measurements. You'll get to see that these are super densely packed, and those are the ones that are inactive. Then you have open regions in your DNA, and those are things like what I'm showing here, right, that are being expressed. You need to be open, and this is what you really care about, which genes will actually come close in space, because now these three can be co-expressed together. The transcription factor can come in and can actually regulate them together. This is what you really care about, how they are organized in space. And that's what we have looked, with also other papers where we just look at the packing and expression, saw that, genes that have transcription factors that can turn them on together, they're actually close in space in these active regions. That if you have clusters, if you look at which genes are together, the ones that are repressed are together, in these clusters, you always have active clusters and repressed clusters, the things that you would expect. Also, what is known is what you just said, these shells. So the more active stuff is actually in average, right? So there is also heterochromatin, so these compressed regions in the center, but there is more active stuff in the center. And in particular at the boundary, you can actually use the nucleus boundary to shut off stuff. So everything that is stuck to the boundary is shut off. So there's really nice kinds of, organization. And we've studied a lot of how it is optimized for function and for expression. And I think there is really nice maps between these two things, and there is still a lot to be done, but there is certainly already a lot of evidence that this is very interesting.
Mm-hmm.
Yep.
Okay, thanks.
Thank you.
Any other questions? Maybe I could come back to mentioning a possible connection that I see to topological data analysis. When you're talking about these higher order interactions that are determined, and when you're looking at the distances between the different loci, I was wondering whether some of the tools of persistent homology might not lead to some sort of extra signature that might be helpful for distinguishing between different cases. Because once you start taking these, one way to encode this information is through something like a filtered simplicial complex, which I'm just mentioning that as maybe an extra tool for describing the structure that you're seeing.
Yes, and I remember like many years ago I thought about this, but in some sense, so I think I don't have the data. In high C, I don't think you have. In imaging, yes, I think this will be really interesting. But like here, I'm not sure, because you would do like this. You guys are the experts, but like. So I don't know if I would have the data to do it, because here I'm exactly measuring like how often do three things come together, how often do two things come together, how often do four things come together, and you would want to know like how persistent are they, right? And so I don't know if you would know better.
Do you have some notion of distance between these loci? I thought that was what I understood, but maybe I misunderstood. Okay.
No, that's what we're trying to reconstruct.
Okay, you're trying to reconstruct from the frequency. Okay.
From the frequency, this is this map that we're just like, that's what we're doing here, right? We're encoding this into distances. But then afterwards you want to reconstruct the whole 3D reconstruction from something like distances to a centromere. Yeah, so we're turning these frequencies into distances from these centromeres. And so that's why I'm saying like imaging data might be very good for doing that, because then you just get to see where these points are, and then maybe there you could use something like persistent homology or something for actually figuring out how close and which ones are more important, etc. But I think it would be really, really interesting to try to connect.
Yeah, to try to extract from this frequency information. Yeah, that's okay. Yep. An interesting sort of inverse problems somehow. Okay.
Yep.
Thank you. Are there any further questions? If not, then I suggest we unmute and applaud to thank Caroline very much for a fascinating tour of mathematics and structure. Thank you very much for having spoken to us today. It was a really great talk.
Yeah, thank you very much for the invitation. It was really fun.
Yeah.
Made me think more about what I should prepare and what I should talk about in terms of geometry.